Hi, my name is Lulu Singer, and this is my independent project for Phage Genomics. So for my project, I wanted to look at the DQ phages. Of the two phages we annotated, I thought Chisana Kitsune was a lot more interesting, and I thought the DQ phages were more interesting than the CZ2 phages. So one thing that really struck me in the 2017 Gordonia phage paper we read was about how they couldn't cluster Gordonia phages the same way they could cluster the Mycobacterium phages. So with the Mycobacterium phages, they could just cluster based on nucleotide identity. So phages that are in the same cluster just have greater than 50% sequence similarity, I think that's the number in the paper. But for Gordonia phages, when they tried to do that, they got a lot of clusters and a lot of singletons, and phages that would have a lot of similar gene content but low nucleotide identity would not be clustered together. So instead, they based the Gordonia clustering on gene content similarity. And for the Gordonia phages, I thought obviously that made sense, but I also kind of wondered, would horizontal gene transfer have maybe a bigger impact on the way the phages were clustered now if they were clustered based on gene content similarity? So the first thing, this is the set of phages I used in my project. So these are actually the phages with the most sequence similarity to Chisana Kitsune, but also these phages here represent all the clusters that are most closely related to the other DQ phages. So for example, all the DQ phages were very closely related to the DX phages, as well as the singletons, claws, and Canter, which is actually a Brevibacterium phage. Additionally, they were all closely related to these three CT phages, but only these three. Also, they were very closely related, or they were related to these D1 Mycobacterium phages. Chisana Kitsune was just related to these two, but Chidi Berry and Gray actually were related to a lot more D1 phages. And lastly, Chisana Kitsune had significant sequence similarity to these DG1 phages, even though Gray and Chidi Berry did not. So to start off with, this is a heat map based on nucleotide identity. So here you can see the DQ phages, and you can see GMA6 does not appear to be as related to the other DQ phages as they are to each other. It actually looks pretty distantly related. In fact, you can see here are the DX phages, and on average the DX phages had a nucleotide identity of about 0.6 with the other three DQ phages, while the three DQ phages had an average nucleotide identity of 0.65 to GMA6. So you can see how it doesn't really seem that related, even compared to how related these DQ phages are to the DX phages. Additionally, this is pretty interesting that these DG1 phages, which are Gordonia phages, share more nucleotide identity with these two Mycobacterium phages than all of the Gordonia phages here. I found that interesting. And then additionally, you can see here that Canter actually has a lot of, has more sequence similarity with the DQ phages than some of the other Gordonia phages, which again is interesting because it is a Brevibacterium phage, and you can see it's really not as closely related to other clusters other than DX. So this is a heat map based off of gene content similarity. As you can see, it does look pretty different in contrast to the nucleotide identity heat map. You can see here that the DX phages now do not look that closely related to the DQ phages, while Canter appears a lot more closely related to the DQ phages than any of the other Gordonia phages. Additionally, the two D1 phages that seemed kind of or relatively similar to these DG1 phages, now they look a lot more similar to them than others. So, all right. So here also, I just wanted to talk again mention how GMA6 is really kind of an outlier in the DQ phages. It's really weird. For example, in the paper, they mentioned that of all the singletons, they all had above 30% of their genome as orphams, so 30% of their gene fams had only been found in them. And I just thought it was interesting that GMA6 did have over 30% of its genome unique to it, which is significantly more than any other DQ phage or even the DX, CT, D1DG phages. And the only other phages that had more orphams, obviously, were the two singletons that came up. So I kind of wanted to talk about how this all can be seen, or how clustering based on nucleotide identity or gene content similarity affects what the evolutionary history of the phages kind of looks like. So, like as I was talking about before, with average nucleotide identity, the DQ phages seem a lot more related to the other Gordonia phages, such as the DX phages, and they seem more evolutionary distant from Canter, the Brevibacterium phage. But because they only had, a certain number of fams in common with Canter. But here, when it's based on gene content similarity, those few fams that they did have in common kind of distort the phylogeny tree more than, just sequence similarity would. So it looks like the DQ phages are more related to Canter than like red wattle hog or Stormageddon, which are the DX phages, despite Canter being a Brevibacterium phage. And as I mentioned before, GMA6 is not super related to the other DQ phages, right? It has 30% of its genome is just orphams. And you can see here how GMA6 looks a lot more closely related to the other DQ phages than it does here, when the tree is just based on nucleotide identity. Additionally, as I mentioned before, these two Mycobacterium phages did have some similarity to these DG1 phages. And while both of the trees kind of show them as being the most closely related, it looks like on this tree that they are less distant evolutionarily speaking than in the nucleotide identity tree. So it looks like they diverged a lot more recently, when in reality we know they probably share gene content because of horizontal gene transfer rather than a shared ancestor. Additionally, I wanted to mention these three CT phages. As I mentioned before, all of the DQ phages appear to share similar sequence similarity with the CT phages, but the three CT phages, those are out of 29 CT cluster phages. So the fact that only those three were similar to the DQ phages might also be an example of horizontal gene transfer, or just some amount of DNA being picked up even if they don't necessarily share a fam with the DQ phages. So when it comes to like the big picture of clustering all the Gordonia phages, it did make sense to cluster based on gene content similarity. Clustering based on ANI would not have, produced any like reasonable tree or reasonable clusters where each phage was in one cluster and one cluster only, and there weren't an insane amount of singletons. But when you kind of look at things on a more granular level, you can see how horizontal gene transfer can distort the results or the phylogeny trees or the clusters made based on gene content versus nucleotide identity. So, yeah.